Welcome to Click for IB Biology. We're practicing our skills of interpreting electron micrographs of cells. There's a PDF of this presentation linked into the description. Our first image is this one here, and this image A is of the nucleus. And in this nucleus, we can identify a number of features. First of all, around the outside, and beautifully shown here, if one looks very carefully, is the double membrane of the of the nucleus labeled B. So, double membrane of the nucleus. This very dense, dark region here is actually the nucleolus. I'll just write this up here. Nucleolus. And the nucleolus, surprisingly, is where our RNA is combined with proteins in the production of the subunits of ribosomes. Very complex process. And then those ribosome subunits are exported through pores in the nuclear membrane, again structure B, into the cytoplasm, where we would recognize them as ribosomes. D is referring to what we would call the nucleoplasm. The nucleoplasm is the nucleus's version of the cytoplasm. And if we look into this region here, we can see that there are a combination of slightly lighter gray colors, and then of course regions like this of slightly darker colors. Now this is the DNA material itself, known as chromatin, and chromatin is actually a combination of DNA and its associated proteins called histones. And where we have these dark regions here, this is very, very dark, dense DNA histone chromatin, which is actually called heterochromatin. And for students already doing protein synthesis and gene expression, this represents the chromatin, or the DNA if you like, which is very dense, very densely coiled, and is not being expressed as a gene in transcription. Whereas these lighter gray regions, we would call that euchromatin, and this is associated with less densely coiled DNA, in which the genes are involved in transcription, or a more general term would then be the expression of the gene. Okay, let's apply this knowledge to our image over here. So this is our test image, and this is where you, the viewer, gets to do your thing. What name would we apply to structure A in this image of the nucleus? Okay, so giving you time to look at that, of course, this, in fact, of course, is the nucleolus. We've got the double membrane of the nucleus around the outside, which, to be honest, I can't see in this image. We've got the nucleoplasm here, but A would be the nucleolus. Let's take a look at another structure. This is a beautiful image of the mitochondria. And the mitochondria, like the nucleus, has two membranes going around the outside. The outer membrane is smoothly going around the outside to give this bullet-shaped structure, but the inner membrane is folded into what we call the cristae, which I'm kind of overlapping here, and we can see these cristae folds. The interesting discovery about the cristae in recent times is that the cristae formation is dynamic. That is to say, it forms and then it breaks down and then it reforms in response to the metabolic demands of ATP production. And If you've studied respiration already, you'll know that there are significant events taking place on the membranes of the cristae. And then in C, I'm pointing to the inner structure. So inside the folded cristae membrane, the contents of the mitochondria are known as the matrix. And the matrix is the location of a cyclic metabolic pathway known as the Krebs cycle, associated with the synthesis of ATP and of the reduction of coenzymes. So something for you to link out to your other studies here. Now, before we leave this image, we have to know these structures here. This single membrane structure here is in fact endoplasmic reticulum, where the endoplasmic reticulum has been cut in cross section or transverse section to show a tubular structure. And around the outside of this tubular structure, the endoplasmic reticulum, are the dots shown here. E, I'll just highlight these darkly stained dots here, and those are the ribosomes. So E and D together make rough endoplasmic reticulum. Let's apply our knowledge. We've got two tests here for B and C in these images. So you can pause the video and work out what is structure B indicating. And I'm referring to this region here. And what is structure C pointing to? And I'm pointing at these tubular structures, which, if one looks very carefully, there are dots along the surface of that. Pause your video, work out your reasoning for these two structures. And of course, B is pointing to the matrix of the mitochondria. We can see the outer membrane here. We can see the folds of the membrane showing the cristae, but the inner content is the matrix. In this image here, we have an image of rough endoplasmic reticulum with the tubular system of the reticulum with ribosomes stuck around the outside. And students may recall that such endoplasmic reticulum is associated with the production of proteins which are due to be exported from the cell, from rough endoplasmic reticulum. Okay, let's move on and look at our next example of a common electron micrograph structure. And already I'm sure as you look at this, you'll realize that this, of course, is a chloroplast. Now, the chloroplast, like the nucleus and like the mitochondria, if we look carefully to the membrane here, we see that we have a double membrane indicated here by structure E. I've labeled that. So we have a double membrane. Now, for those of you doing higher level biology, the double membranes of the nucleus, the mitochondria, and the chloroplast, you should be thinking of endosymbiont theory, which was proposed by Lynn Margulis to explain the evolution of eukaryotic cells. I'll leave you to follow that line of thinking. Let's return to our chloroplast, the site of photosynthesis, and not going through in letter order, but C and B are referring to the membranes. Now, B is a thylakoid membrane, and C is actually a stack of those membranes called a grana. And these membranes are associated with having protein pigments rather embedded in them, which are chlorophyll, which of course absorb light energy, turning light energy into chemical energy in the form of electrons. Those electrons are used in zone A. Zone A is called the stroma, and the stroma is the location of another cyclic metabolic pathway, which is known as the Calvin cycle. And the Calvin cycle is where the chemical energy produced by the trapping of sunlight energy on the thylakoid membranes is used to synthesize sugars, simple sugars. That's it, A. Now, the final feature on this is structure D, which actually the idea that simple sugars are combined to form hexoses, 6 carbon sugars. In turn, those are combined together to form chain molecules, which are examples of these white regions here, which are actually starch. These are starch granules inside the chloroplast. And before we depart, it's worth noting in this diagram, this structure here, which is the classic image that we would see of a plant cell wall. So let's test your knowledge. I'm going to ask you, what is the structure here labeled D, and what is its function? So pause your video, work out the answer. And of course, the answer to that is that D is pointing to the grana of the thylakoid, and that is the location of chlorophyll, which is trapping the light energy and turning this into chemical energy, which of course is the initial stage of photosynthesis. Let's take a look at our next micrograph. And this micrograph here is a development of the previous image. And here we have the plant cell wall. So this is the cell wall structure, usually characterized by the quite clear zones down the center of two very parallel lines like so. So of course we are looking at a plant cell structure. On this side and this side of the cell wall is the cytoplasm, and then this membrane here, and indeed this membrane here, going into zone C, I guess we could put zone C as well. These are the membranes of the vacuole, which are known as tonoplasts. But the main feature I wanted to look at was that of the cellulose cell wall. We come across the cell wall in a number of parts on the course, very particularly when we're looking at xylem structure in plants known for their conduction of water and minerals. The cellulose cell wall also forms things like the phloem sieve plates shown here, showing the pores in the end walls of the phloem tissue. And then this very interesting structure, which is that in this image here, we have the cell wall is here, which is structure D. But if we look carefully with the cytoplasm here and here, there is a cytoplasmic connection between the two cells through a structure which is called a plasmodesmata. So the plant cell's cytoplasm is connected to each other, which is very atypical of cells, and molecules of sufficiently small size can pass between those through that cytoplasm. And one in particular, of course, is the movement of water. And the movement of water from the cytoplasm of the cell on the left-hand side here to the cell on the right-hand side here through the plasmodesmata is known as the symplastic pathway. And this will come up later when you are looking at aspects of plant transport. Okay, so having looked at this, let's pause the video and identify structure E in the diagram and what is suggested by structure F in this electron micrograph. Stop and pause your video now and work out your answer. And of course, the answer to this is that E is the cellulose cell wall of a plant. So it's made of cellulose and it's in a plant cell. And F is the idea that with the cytoplasm here and the cell wall here, there must be a cell membrane running up here, which is being pressed out against the cell wall due to the turgidity of the cell, and that would be structure F, which of course we can't really clearly see in this image. And so my advice is that you go to something like Google electron micrographs, and practice your interpretation of the images that you will see there. Further information on organelles and cell structures are shown in these video links here back to the main Click for IB Biology site. Please give this a thumbs up, give it a like, and subscribe so that you get notification of new videos.